Hello and welcome. This is Bhaskar Napte from Pharma Growth Hub, and I am sure today's video will certainly help you while investigating a failure in the precision study while conducting the analytical method validation. So let us understand, what is that ICH guideline which help us in providing the guidance on validation of the test procedure, and that is none other than the ICH guideline Q2R1, that is validation of analytical procedures, text and methodologies. Further, let us understand, before we move on to the likely causes for the failure in the precision, first understand what is mean by the precision according to the ICH guideline. So the precision of an analytical procedure expresses the closeness of agreement or degree of scatter between a series of measurements obtained from multiple sampling of the same homogeneous sample under the prescribed conditions. So you are going to analyze some number of samples as a part of precision study, and you are going to assess how the results are closely associated with each other. If they are closely associated with each other, if they are close from each other, then you can say, okay, I now got the precise result. But in case if the result are not close to each other, they are away from each other, then you can say now I got the imprecise test result. So that is the meaning of the precision study. So the precision study can be conducted with the help of 9 determinations, right? By covering the specified range for the procedure. Means what is the range of your analytical procedure that you can think of, and then the lowest concentration can be your lowest range, and the highest concentration for the precision can be highest concentration in the range study, and the third concentration can be or probably the 100% or the specification level. So that way you can understand the three different levels, and each level have to have three different determinations. So on all together you will get the 9 determinations. So this is the one way of conducting the precision study. But in case if you do not want to conduct the precision study at 9 determination, you can conduct the precision study by determining the 6 determinations, but now at the 100% of the test concentration or at your specification level. So this is how the precision study can be carried out. So now let us understand, what are the likely root causes for the failure in the precision study. The first and foremost, a very important likely cause can be the non-homogeneous sample. See, in case of precision study, you are trying to understand how the results are closely associated with each other. For example, you are determining assay for a paracetamol tablet. And if you get the assay values like 99%, 100%, 98%, 99.5%, now you can say my results are very, very precise or closely associated with each other. In case if you have the homogeneous sample, the chances of getting the closely associated results are likely. But in case if there is no fault into your testing procedure, however, the sample in the hand is not a homogeneous. It is a non-homogeneous. Some of the tablet contain 110% paracetamol. Some of the tablet contain 99% or 90% of the paracetamol. So if you happen to choose this different content tablets, don't you think that your assay result can be sometimes 110, sometimes can be 100, and sometimes can be 90% also? Now, is this the fault of your testing procedure or the sample in the hand? Right? The obvious reason is what now? The sample is non-homogeneous, and because of that, you are not able to get the precise result. So having a non-homogeneous sample can certainly end up getting the imprecise testing result. The question could be, in case if I have the non-homogeneous sample, can I do something out of that to come out this challenge? Yes, you can create the sample by taking appropriate amount of the placebo and the paracetamol API. With the help of placebo plus API, you will be able to uh, prepare the sample which can simulate your drug product. So that is how the non-homogeneous sample could be the reason for your imprecise result, and you will be able to come out that by making the sample by using placebo plus API in case of the drug product. In case of, let us say, drug substance, likely chances of a non-homogeneous sample is very low, but still in some case of if your sample is coarser crystalline material, that can be a reason. So you can crush the sample into a fine powder and probably the sample can become the homogeneous after that. So use the homogeneous sample to assess the precision of the testing procedure. The second root cause for imprecise testing result could be the incomplete extraction. See, now this is very critical part where your diluent selection at the stake. So if your diluent is not able to extract the analyte from the sample matrix, then in some preparation you can have the 90% extraction resulting into a 90% assay. In some preparations you have the 100% extraction and then you get the 100% result. So the analyte was present in the 90% sample also, but this 10% analyte was not extracted out of your diluent system, and that resulted into the 90% assay. So you can end up getting the imprecise testing precision result in case if your diluent is incapable of extracting your sample or your analyte. So understand whether I have the right amount of the extracting solvent. Am I using the right extraction technique like sonication versus shaking? Do I need to give some thermal treatment so that the extraction become efficient? Am I using the right diluent system and extraction technique is the question. So this could become the reason for the imprecise testing result. So make sure your diluent is appropriate, make sure you're using the appropriate extraction technique, and then probably you'll be able to get the precise analytical result. The third important root cause for imprecise testing result can be your weighing error. So for example, you are having a powder or a sample in the form of a powder, and that powder consists of a fines and a coarser material. So imagine if you collect this powder into a butter paper and you are in the process of now transferring this powder onto the balance pan. So you have to give some, a little bit movement to the butter paper so that the powder can slowly get transferred onto the butter paper. So during this process, the segregation take place. The coarser material can move faster and the fines present into powder will get stick onto the surface of the butter paper. So just imagine in case if the analyte concentration into a coarser part is higher, you may end up getting the higher result and vice versa. So the segregation can happen during the weighing and this can further result into the imprecise analytical results. So how you can come out of this challenge? It's very simple. You can grind this powder into a fine form so that there is no difference between the coarser or there will be no presence of the coarser material present into a sample. So during this weighing, the segregation will not take place and you will not get the imprecise testing result. The another important part is the crushing of the sample. Sometimes in case if you have a coarser sample or in case if there is a tablet, you need to crush it into a fine powder. So that crushing is also very crucial. And as I explained that if you have the fines and coarser materials, the segregation is going to happen and that can result into the imprecise testing result. The third error that is possible is the during weighing is the hygroscopicity of your sample. See what happens, you are taking a sample, you are in the process of weighing now, and the sample is highly hygroscopic in the nature. So if your atmosphere has the 70-80% humidity, now during the weighing, the sample can pick up the moisture and that will create the error during the weighing, isn't it? So if you allow more time for weighing, the more amount of moisture can get picked up, and that will result into the some else result. If you measure the sample in a short period of time, weighing you do in short period of time, then the little amount of moisture will get picked up, and that will again result into the different result. So the hygroscopic material can become the reason for the imprecise testing result. In such cases, you have to take utmost precautions. You need to control the humidity maybe below 40% RH, and that will become the suitable environment for the weighing. So I think the third probable reason could be the sampling weighing error. Then the next root cause for imprecise result could be the instrument variation itself. For example, you are using a chromatographic system like HPLC or gas chromatography, and if there is a injection to injection happening, injection to injection variation happening, though you supposed to inject, let us say, 20 microliter solution, but your system as itself is giving some error. Sometimes it is injecting 18 microliter, sometimes it is injecting 22 microliter, and that will certainly result into the imprecise analytical result, because the response will get vary according to the injected volume. And then you will say, now my testing result is imprecise, but the root cause is what now? The injection to injection variation. Sometimes the injection volume can also get altered due to the air bubble present into the syringe. So make sure that your syringe is free from the air bubble, so that the imprecise result will not occur out of the instrument variation. And there could be some more reasons. You can understand that based on to the hypothesis part. Then the next root cause for imprecise testing result could be the unstable solution. But before I move on to the unstable solution part, let me briefly introduce myself to all of you. Hi, my name is Bhaskar Napte, and I have almost 20 years of experience into the pharmaceutical industry. I worked for top pharmaceutical companies like Dr. Reddy's, Apotex, Glenmark, and US Vitamins. And after that, I have found the a platform which is named as Pharma Growth Hub. And this platform is helping many pharmaceutical professionals just like you to get the absolute clarity on various topics, to help them to accelerate their career growth, giving a proper support and guidance onto the job search strategies, etc., so that you will not become helpless into your growth journey. So in case if you are interested to join the Pharma Growth Hub, there is a never before offer. You can find the details of the offer into the description. So check the offer and join the Pharma Growth Hub today itself. Thank you so much for allowing me to introduce myself. So as I said, the next important point is the unstable solution. So it's very obvious, isn't it? One can completely relate that in case if the solution itself is not stable, I may get the reduced concentration in case of, let us say, assay, or the impurity concentration can get increased over a period of time because it is getting generated from the analyte or the API. So unstable solution also can become the reason for the imprecise result. So how to tackle this unstable solution? You have to define the uh, usage period for your analytical solution, that within 24 hours the analytical solution has to be tested, and hence you will not get the imprecise result at least out of the usage of the unstable solution. Then the next point can be the inappropriate filtration used. See, the filtration is very much important in case of the complex sample matrix, especially for drug products containing the polymer based excipient. So this filtration techniques needs to be validated. So what is suitable filter paper? Is it nylon filter paper? Is it a PVDF or is it a PTFE that has to be used? And on top of that, what is the saturation volume? Now the saturation volume is what? How much of the analytical solution to be passed on the filter paper so that the filter paper will stop further absorbing your analyte. See, because every filter paper has this absorption and adsorption capacity. So whatever analyte is going through the filter paper will get bind onto the filter paper to certain concentration level. And that concentration is called as what? The saturation concentration. And once that saturation concentration is reached, then the further analyte will not get bind by the filter paper. So you need to understand how much is the filter saturation value. Is it 1 ml I need to pass on the filter paper? Is it the 5 ml that I need to pass on the filter paper? And once that has been achieved, then further analytical solution can be collected for the quantification. So understand what is the filter paper to be used and how much is the saturation volume. Then the next root cause can be the interference from the sample matrix itself. Now this is very much a fundamental need of analytical method, that it must be a selective or specific in the nature. That in case if there is a sample matrix available, but that sample matrix should not interfere in the quantification of the analyte. So in case if there is an analyte, if there is a sample matrix which interferes with your analyte, then certainly that response is not going to be consistent, isn't it? And because of that, you can certainly have the imprecise testing result. So please check your specificity of a test procedure before you comment on to the root cause for the precision failure. And then last but not the least, the poor analyte response. See, our analytical techniques has the limitations. The response will be accurate and precise after some response. In terms of the absorbance value, in terms of the peak area, we have some limitations. All responses cannot be quantified precisely. And you need to understand whether the response is suitable in terms of its precision, in terms of its repeatability. So understand in case of HPLC, how much should be the peak area below which I should not go. And if you are below that, especially in case of trace level impurities like nitrosamine, mutagenic impurities, the concentration is going to be much, much low. So please make sure that you are choosing the concentration in such a way that the response for analyte falls in the range of the precision. So that is the advice, that analytical response also has to be checked. So thank you so much for watching this video, and I will meet you soon with such kind of informative videos. Till then, keep learning. Bye bye.